Thanks everyone for coming to my talk today. I'm going to talk first about kind of the state of the knowledge of trunk diseases in nursery stock. But first I'm going to give you a little bit of an introduction for those of you who aren't familiar with the study system. Every vineyard in California eventually becomes infected by one or more of these trunk diseases. I refer to them collectively as trunk diseases in part because they often occur in mixed infection. You'll find more than one of them typically in a vineyard, in an individual vine, and even sometimes from an individual wood canker. You can isolate more than one of these pathogens. These are one of the main reasons that vineyards are replanted on a premature basis in California, and we find the same situation happening in every major grape production area of the world. The names of the diseases have changed over the years. The names of the pathogens have changed over the years. We've learned about new pathogens. A lot of this work was started by Doug Gubler, who's an extension specialist at UC Davis. He retired recently. Probably you've seen him speak about these diseases before. But for many years, we thought it was just one trunk disease, Eutypa dieback. And Doug had been isolating other fungi for years and years. And there's a lot of researchers thought, no, those must just be some contaminants. It's some other fungus. It's not as important as Eutypa. And Doug did some real pioneering work, and it was recognized for this worldwide for acknowledging that these other fungi were not just contaminants. He did a lot of careful studies in the greenhouse and in the field to demonstrate that they were indeed pathogens. Some of the things that these fungi share in common, even though they're not genetically related to each other, is that they all cause chronic wood infections. And the infections are internal and they're slow to develop. Mark was just talking about how sometimes you have a virus infection in the rootstock, and it takes, multiple years for it to move up into the scion, into the canopy, and to show some sort of noticeable symptom. It's very much the case with the trunk diseases. They're very slow to develop, and the internal wood symptoms that are shown in these pictures don't typically appear externally. You can't find them until you saw through the vine. So it's tricky to diagnose these diseases until they're already developed. We have the wood canker producing diseases, which I call the dieback type diseases. That's Eutypa, Botryopsferia, and Phomopsis diebacks. And then there's ESCA, which is also commonly known as measles or black goo. It causes a different type of wood symptom. This is a cordon where the bark's been scraped off, and the black lines are one of the symptoms of ESCA. And then there's another set of ESCA pathogens that cause a totally different wood symptom, which is a wood rot. It's technically called a white rot, and it's a type of wood that's visibly rotted. The texture is really soft. It's very clearly dead. Sometimes it's at the center of the vine. Other times it's, closer to the edge. And I'll talk a little bit more about this later. As I said earlier, Eutypa dieback was the trunk disease we thought was the only one for many, many years. And about 20 years ago or so, we realized there were other diseases. I'm going to talk a little bit more about those other diseases today because Eutypa dieback is one of the trunk pathogens that hasn't been found to contaminate nursery stock. It's the other ones that seem to be more common in contaminated nursery stock, especially ESCA. So the ESCA pathogens in mature vineyards, they cause these very characteristic interveinal symptoms. We call it an interveinal leaf scorch. In red cultivars, you get these red sections developing in between the leaves, and that gives way to a scorch. The dieback type diseases cause these dead spurs, stunted shoots that eventually turn into a dead spur, and that's what you see in a mature vineyard. But these diseases in nursery stock don't necessarily cause these symptoms in young plants. So what you see in young plants from the ESCA symptoms, some of the dieback pathogens, and the blackleg pathogen, which I'll talk about a little bit later, is you see cuttings and bench grafts and rootings that just do not emerge from dormancy. They just die while they're in cold storage. For those that don't die, when they're contaminated with these trunk pathogens, sometimes they push shoots and roots, but those tissues never grow well. They're always weak plants. We get poor graft take. That's a common symptom of trunk pathogen infection of nursery stock. You will sometimes see the wood symptoms. So if you cut into an infected cutting or a bench graft or a rooting, you can sometimes see a canker or the black spotting that you see from the ESCA pathogens. Sometimes it's completely asymptomatic, though. And the severity of those wood symptoms doesn't seem to be consistent with certain rootstocks or certain pathogens, just as we find when we inoculate plants in the greenhouse, looking at different cultivar susceptibility or rootstock susceptibility, we see certain pathogens infecting certain rootstocks or scions more often, but no one is the worst combination. The dieback type trunk diseases, these are some pictures of young plants, cuttings, and some plants that we've inoculated too. I want to mention just a few important things about why the dieback type pathogens can be a threat to young plants, to nursery stock. So this is on your left. There's this is a cutting where I've scraped away the bark, and you can see there's a big canker there. When you cut through it, you see a wedge-shaped canker with some healthy tissue, mostly. And I want to mention that some of these pathogens can sporulate after they infect young plants. So this is the Phomopsis dieback pathogen. And these little black dots, there's a Sharpie here for size reference, but they can make spores on these tissues within 3 months. So if you could get an infected plant in the field, theoretically, and that could, if the pathogen could become established and start making spores, you could collect an infected cutting. That's one source of infection. The ESCA pathogens and also the blackleg pathogen can also, they have been found in nursery stock around the world. Not many studies have been done in California. Most of the work I'm talking about has been published in Europe. I put the names of the fungi here. You don't need to memorize them. They seem to change all the time, especially the blackleg pathogen. For many years, this was called cylindrocarpon root rot. Now it's neonectria. Anyways, it's the same fungus. But in cross-section, when you cut through infected plants, they have this centralized necrosis. So all that tissue in the center is dead and infected. And it's darkened in part because of the plant trying to make phenolic compounds to kill off the fungi, and also because we think some of these fungi, probably this one more so than the ESCA pathogens, can degrade the wood to some extent. So a lot of people are trying to figure out, in the nursery production process, where do these pathogens come from? Which step is most important? And certainly, vines get, just as in regular vineyards, in nursery production blocks, vines can become infected in the fields. These fungi make their spores with rain, and when vines are pruned, the spores can infect the pruning wounds. So you can have that happen in any vineyard, whether it's a rootstock production block or a regular wine grape vineyard. Another stage of production where plants can become infected is when they're being rooted in field soil. Some of the trunk pathogens, and I've listed them here, do appear to have a soil-borne stage, some sort of spore they make, some sort of structure they make that can survive in the soil. And that's true for the ESCA pathogens and that's true for the blackleg pathogen. Of course, the frequency of these root infections, or if they're infections that happen through the wound at the base of the cutting or bench graft, that there's no data on that. All I can tell you is that when we do inoculations with these pathogens to plants in the greenhouse, we infect the plants by taking a suspension of spores in water and pouring it into the soil, and the plants can become infected that way. So, and this has been demonstrated in the field on a limited basis. So theoretically it can happen. How often it happens, nobody can say. And then the other way that nursery stock could become infected in the field, if it's being rooted in the field, is through wounds. So there's a wound at the top of the cutting, there's a wound at the base of the cutting, and those can get infected by these different dieback type pathogens that have splash dispersed spores. So nursery stock that's rooted in potting mix and maintained in a pod as a potted plant, after the cuttings are taken in the field, obviously wouldn't have that infection source from the soil. So that would kind of cut that whole potential out for infection. So given that there are trunk pathogens that have been found to some extent in some nursery stock studies, what can the nurseries really do to stop this from happening or to minimize the infections? And one thing they can do is what you as growers can do. They can put on pruning wound protectants in the mother blocks where the cuttings are being taken to minimize infection of those mother vines. Because if those mother vines don't get infected, there isn't a source of inoculum locally within the vineyard to infect the cuttings. They can use proper sanitation in the mother blocks. And what I mean by sanitation is pulling out vines that are clearly infected and getting them far away from the vineyard, especially in the winter time. Cutting out, if they're retraining vines like you do in the vineyard from the base of the trunk or from a new cordon, cutting out the infected wood and getting that out of the vineyard in a timely manner. They can also, after they've taken the cuttings, when they soak them or after they soak them, they can use various fungicide treatments that could kill any spores on the surface of the bark of the cuttings. They can also use the callousing procedure, which is done under very high humidity, warm conditions, which are great for a lot of fungi. They can just ensure that they're using a sterile callus mix, making their callus room very clean and tidy, keeping that free of potential sources of infection that come in on one cutting. If you have a few cuttings that are infected and then you put those plants, bunch them together with lots of other plants, and then you have them in a hot, humid environment, the potential for spread of infection could potentially be high. So using a sterile callus mix and having a really clean callus room could minimize that stage of disease spread. And then obviously doing everything they can to plant into disease-free or pathogen-free soil out in the fields when they're rooting plants. So for you as growers, what can you do to minimize the risk of having nursery stock come in that's infected? You can always visibly inspect it, and I'm sure all of you do your best to try to check the plants, make sure they look healthy. You can cut through them. You can look for the wood symptoms. And then I mentioned also ensuring proper soil drainage, because this year I saw because we had record rains this past winter, I saw a lot of blackleg this year in mature vineyards, in young vineyards, and certainly the combination of the presence of the pathogen and poor soil drainage together can lead to disease expression. Disease, especially with the trunk pathogens, and this kind of gets to maybe one or more of the questions that members of the audience had for Mark just before I talked about, Is there some sort of stress that leads to disease expression? That could be the case too with some of these trunk diseases in nursery stock. It's the combination of the presence of the pathogen and then the predisposing climate conditions or soil conditions that lead to disease outbreak being a problem. And then another thing I saw a lot of this year was the apoplectic phase of ESCA. I showed you the symptoms of that earlier, these very characteristic intervanal leaf symptoms. There's another set of symptoms that ESCA can express when there's incredibly hot days, weeks, months during the growing season, where a healthy-looking vine can completely wilt and die. And you can see kind of a row of three vines in a row here. The one on the left, next to my dog, is dead. And then the other two are kind of like on their way towards wilting more and dying. This year, some combination of climate symptoms led to many instances of the apoplexy symptom of ESCA. It doesn't mean, though, that this disease wasn't present in these vineyards before. It was there for years. It may or may not have been showing the typical symptoms of ESCA, but for a number of reasons, we had apoplexy symptoms this year. And then another thing I want to mention that we've been working more with in our lab is there are other fungi that are involved with ESCA, other than the ones that cause the black lines and the spotting and the wood, and these are the wood-rotting fungi. And here's a vine that has some fruiting bodies coming out of it. This is a species of fungus called Sterium, and when you cut through beneath these, you can find this wood-rotting symptom. And this vineyard happened to have been grafted over. I've seen a fair number of grafted over vineyards with no problems at all, and some with these ESCA diseases. But in any event, we've been trying to pair up in the greenhouse these wood-rotting fungi. One of the species is called Fomidoporia, with a typical ESCA pathogen, one of which is called Pheomoniella. And when you inoculate them alone to the trunk, to the stems, the woody stems of potted plants in the greenhouse, you get some sort of wood symptom from both alone, either alone. So from the wood-rotting fungus Fomidoporia, we get intervanal patches of drying, of scorched patches of the leaf. And then with Pheomoniella, we get an intervanal yellowing. And then when you combine them together, you get a combination of those symptoms that looks a lot like what we see in the vineyard. So it seems like these co-inoculations, we call them, lead to kind of a synergistic effect on disease severity or symptom development. And it's a phenomenon that's been studied widely in plant pathogenic viruses, grape viruses too. It's like one virus alone isn't that big of a deal, but when you have two of them together, they have a synergistic effect and they cause severe symptoms. Well, this might also be the case with the trunk diseases, and sure enough, we find them in mixed infections. We find multiple pathogens in the same canker, and those inoculated alone to vines can cause problems alone, but when they're together, they cause more severe symptoms. And then I noticed here on the timer, I just have a minute left. Okay, so I'm going to go very quickly just through, just reminding you that, once you have a young vineyard that's planted, whether it's, with hopefully what's healthy material, it is important to start managing for trunk diseases before you see any symptoms. Too often, we find that, We don't do anything about a problem until we see the symptoms happening, and then it's kind of too late because, as I've been saying all along, these fungi cause chronic infections, they get into the wood, and they're slow to develop. So the idea is to be as proactive as possible. You first of all have to assume that it is going to happen, but then second of all, certainly there's something you can do about it, and you should, but don't wait. The most damaging aspect of the trunk diseases, I think, are the dead spurs. If you have a dead spur, you're losing shoots. For every lost shoot, you're losing fruit clusters. And so as these infections accumulate in a vine over time, your yields go down and down and down. A study that was done in the late 80s and early 90s on what was a cultivar that was once planted widely, columbar, which is very susceptible to the dieback type trunk diseases, showed a very steep increase between year 10 and year 15 in the percentage of vines in the vineyard that had symptoms, whether it was a dead spur or a stunted shoot or a wood canker. Vines aren't more susceptible during this time, but just as Mark was explaining about the virus is taking a number of 2 to 5 years to show some symptoms, it's certainly the case with the trunk diseases. The infections are starting when the vineyard's young, long before year 10, but it just takes multiple years to see any symptoms appearing. So the idea is to prevent them when the vineyard's young, long before you see any symptoms, because if you don't, you have to use what I call the post-infection practices, and those tend to be a lot more costly. For prevention, we recommend 2 approaches, either delaying pruning to as late in the dormant season as possible. This is when the risk of infection is lower. And also another approach is just to prune when it's convenient for you and put on a pruning wound protectant to minimize the risk of spores infecting pruning cuts. And there are a number of different materials that have been evaluated for that. I'll just go quickly through these different approaches. A few studies have been done, very few studies have been done on pruning wound susceptibility, but they tend to have some consistencies in them. And one is that if you prune in December, the wounds can stay susceptible to infection for up to a month. Whereas if you prune in late February, early March, they can heal up in a matter of a day or 2. So you should prune according to that general pattern. Certainly it can change from year to year, and we don't know exactly what it is that makes vines heal up faster or slower. That has to do with the season, kind of the dormant season moving on. But the cold temperatures, the rains, those definitely contribute to the spread of spores and the infection of pruning wounds. The pruning wound protectants that are labeled for dormant season applications against trunk diseases are Topsin M. This one's the pruning wound protectant that's been studied the most against the broadest range of pathogens and in the most different studies done on different continents. Raleigh is labeled for dormant season applications. It's been tested in maybe 2 different studies. One says it's good, one says it's bad. B-Lock, or as a boric acid paste, that's been evaluated against Eutypa, not against the other trunk pathogens. And Vitaseal's been evaluated by Doug Gubler's lab. And Topsin and Raleigh are fungicides, so the idea is that the spores will germinate on the wound. They don't keep the spores from germinating. The spores will germinate. But those 2 fungicides keep the fungal filaments from growing. So the spores could germinate, but the thought is that if you put this material on, it'll keep the fungus from growing well. Boron is a chemical barrier, just like the fungicides would be, and Vitaseal is thought to be more of a physical barrier. The whole point, though, with these products, regardless of which one you use, is that you get it on before the rain, because the rain is really what brings out the spores. And I put this slide up here to remind you that, rains also dilute these products. So if you put them on early in the dormant season when the period of susceptibility is very long, you might have to do additional applications when there's more rain. And we have a study we started with pruning wound protectants in table grapes. Last winter was our first winter, and we tested, because we had such a rainy winter, we had the opportunity to test multiple applications. And we did indeed find better efficacy with 2 applications for some of the products we used rather than a 1. The idea being that if you put it on and and it rains, it's washed off the wounds or diluted, then those vines might still be susceptible, those wounds might still be susceptible. Putting on another application can get them through that full susceptibility period. Our trial that we're testing fungicides on in table grapes that I just mentioned is using a tractor to do tractor applications. All the studies that have been done on pruning wound protectants, with the exception of one I could think of that was done in Australia, use hand, painted on pruning wound protectants. And we think it's important in our lab to actually do what most growers are doing, which is spraying these on to evaluate the efficacy of the products. Delayed pruning, hand applications of Topsin have been tested in different studies against the Botryopsphera diebac, Eutypa diebac, and ESCA pathogens. And you see a big range of efficacy for some of these techniques, like delayed pruning for the ESCA pathogens. In one study year, it protected 28% of the pruning wounds only, and in another year, it was 87%. There's a really broad range of efficacy for some of these approaches against some of these pathogens in some years. For Topsin, the ESCA pathogens, it wasn't found to be real effective. Hence the combination of Topsin plus Rally. Rally has been shown to be effective a little more effective against the ESCA pathogens. So we still have a lot of work to do with these approaches as scientists. But the most important thing I can tell you is, you probably have more than one pathogen in your vineyard. The most important thing you can do is start using these techniques, one of them or both of them, when the vineyard's young. When the vines are trained up and you're starting to harvest from them, maybe at year 3, at the latest, before if possible, start recognizing that wounds are wounds, whether they're training wounds or pruning wounds, they can get infected during the dormant season. The sooner you pick one of these approaches and use it religiously every year, the better. We've done some studies with the economics of trunk disease management that you can find out more at our website, treeandvinetrunkdiseases.com. or .org rather, and our economist Jonathan Kaplan, he came here last year to speak. This is something him and one of his students developed. And then also on our website we have more details about the management strategies, so you can read more about that there. And then lastly, I just wanted to acknowledge some of our funding sources, put our website up there, and thanks very much for your attention.